Okay, so here's what we've got, guys. We're going to start the biology unit today. We're going to talk about the cell theory. All of our units kind of start out with a little bit of history, okay, of that particular branch of science. We started out in chemistry with the structure of the atom. That's the most basic part of chemistry. Well, for this one, we start out with the cell. It's the most basic part of biology. Okay, we're going to talk about kind of who contributed to our understanding of the cell and that kind of thing. We'll also look at how a microscope works, because on Monday we will be going into the lab using the microscopes, looking at some microorganisms, not live ones, but okay, we'll be looking at them under the microscope and drawing some lab diagrams and things of that nature. Okay, so we got to cover a few things to do with that. Done. We'll look at living organisms, but we won't be like, I should say look at them. We'll talk about them, discuss them, but the only living organism we'll actually do a lab with is a plant. Okay, we don't. You don't do any dissections in Science 10. No, underneath the microscope, no. Reason being, it's really difficult to actually look at microorganisms when they're alive because they're actually quite fast. I know when I was in university, we did a lab where we had to feed paramecium, and you have to actually put glycerin, which is like syrup, on the slide to slow them down. Otherwise, they just zip back and forth, and you miss everything. Okay, so no, the ones we will be looking at are preserved specimens that are stained and brightly colored as a result, easy to see. Okay, we gotta build up to that, right? You gotta have good microscopy skills before you go looking for, it's like when you learn to shoot, you don't start with a moving target, okay, generally. All right, so we want to know the points of the cell theory and how they relate to living organisms. Okay, that's something that we will come back to over and over again in this unit. Okay, that is probably one of the most overarching concepts in the biology unit is here's what an organism is doing and here's how it relates to the cell theory. Guess. Okay, so really important that we understand that. Okay, we need to know who contributed to the development of the cell theory. Somewhat important. Okay, and recognize the implication of the similarities between plant and animal cells in terms of evolutionary relationships. Okay, the fact that they have so many things in common tells us something important. Okay, and that's what we're going to look at. All right, so the picture we have here, simply a diagram of a plant cell. All right, and plant cells contain certain organelles like a nucleus, okay, or chloroplasts, or mitochondria, okay, or a water vacuole, okay, and endoplasmic reticulum, and Golgi apparatus, and things like that. So they got lots of things in there, and each one has a specific job. We'll talk about those specific jobs Thursday and Friday. All right, so the cell theory has 3 points. Okay, the first point of the cell theory is that all living organisms are made up of cells. Okay, some are unicellular and some are multicellular. This is the point of the cell theory that separates living organisms from, let's say, chemical reactions. All right, so what are some other things that a living organism should show, or characteristics a living organism should have? Okay, so exchange of gases, because not all organisms actually physically have lungs and breathe, or gills and breathe. They may just exchange gas back and forth. Yeah, generally, from what we know, made of carbon. Yep. Not all things move. I mean, plants are sessile, right? They don't. They grow. Yes, growth would certainly be some characteristic. Okay, reproduce. Okay, also a good one. Do they use energy? Okay, do they produce wastes? Yeah, okay, all things produce wastes. Plants produce waste. Their waste is just useful. It's oxygen. Okay, that's the waste gas of plants. All right, so things like that. Now, here's the thing. All of those things we listed, okay, exchange gases, okay, they use energy, they make waste, they reproduce. All right, if those were the only characteristics of a living organism, then fire is a living organism. Because fire uses energy, fire produces waste, fire grows, fire reproduces. Okay? A spark from one part of the fire can start a new fire. We'll count that as reproduction. I know that's technical, but okay, it's technically true as well. All right? The difference is Fire is a chemical reaction. Fire is not made up of cells, and it is the presence of cells that is the defining characteristic of a living organism, and that's where the first point of the cell theory comes in. This is the point that says these things are alive and these things are not. Okay? Now, there are some organisms that still kind of flirt with that line. Okay? A virus flirts with that line. A virus is not technically a true living organism because a virus does not have cells. Okay? It is not a cellular organism. It is a strand of DNA surrounded by a protein shell, and that is all. It cannot reproduce on its own. It cannot metabolize food. It does not produce waste. It doesn't do any of those things that we said a living organism does. It is an obligate intracellular parasite. Okay? It gets into your cell and hijacks your cell and forces it to make copies of itself. Right? That's how a virus makes you sick. Okay? I mean, you've all had this experience, right? You go to bed and you got that little tickle in your throat. You're like, oh, I think I might be getting sick. And the next day you wake up and you're like, I'm going to have to get better to die. Okay? Like you feel awful, right? It happens really fast. A virus can take over your body really fast. One virus in one cell in your body can make 1,000 copies of itself. Okay? Sorry, it doesn't make the copies. Your cell makes the copies. All right? Because what a virus does is it takes its DNA and splices it into yours. Okay? And what that does is now your cell, when it goes to make something, whatever your DNA is supposed to tell it to make, it doesn't tell it that anymore. It tells it how to make a virus. Well, your cell's dumb. It doesn't know any different. Okay? It just goes, oh, here's the instructions. Send those to the factory and we'll build it. Okay? Well, that's what happens. Okay? And it builds a virus. And then the cell goes, hey, didn't we ask for that other stuff? We didn't get what we asked for. Send that thing back to the factory and tell them to make another one. What do they make? Another virus. And it just keeps going. Because the cell keeps asking for the thing it was supposed to get, never gets it, keeps building viruses. Pretty soon, the cell explodes. Okay? It literally pops, and all the viruses get out. And each one of those viruses goes and infects another cell. Okay? So it doesn't take long for a virus to really get going in your body. And they're hard to find because they're really small. Okay? Your body uses its immune system, so your white blood cells, killer T cells, okay, things like that. But what it does is it kills the cells that are producing viruses. That's why you get so weak when you have the flu. Your body's actually destroying its own cells to kill the virus because it has virtually no chance of finding a free-floating virus in your bloodstream or intracellular tissue. So what it does is it looks for cells that are behaving abnormally or that have a viral marker, and it kills them. Yeah, they basically eat it. Okay, certain types of white blood cells do it a different way. Some of them inject a toxin in that destroys the cell, and others just eat the cell. Depends on what kind of... No, because it digests, actually physically digests the virus. Yeah. What's that? Well, no. Some viruses can withstand the conditions inside of your stomach. Okay, yeah. You don't. You get more red blood cells if you're athletic. If you're training, red blood cells carry oxygen, so your body makes more if you force it to. Okay? By training, it requires more oxygen to be delivered, so it makes more red blood cells. Okay? That's why people like Lance Armstrong and guys like that are cheaters. Okay? They'll actually remove their own blood. Okay? Freeze it. Make more, like their body will reproduce to reproduce the blood to make up for what was lost, and then right before a competition they'll inject the blood that they took out. So they have extra blood now, right? Way more red blood cells so they can carry oxygen way better, which means they have far more stamina than everybody else. Okay? Oh well, yeah, it's totally cheating. It's blood doping. Okay? It's totally cheating. Yes, because there's certain levels of red blood cells that are normally present even in an elite athlete. Okay, when they do that, the numbers are off the charts. Okay, they know right away that this is an abnormally high number of red blood cells, and yeah, then you're. Well, no, they generally just break down. Your body goes, what the heck? We got way too much of this stuff, and it just breaks them down. Okay, so yeah, but the thing of it is you know, it sounds like, wow, that'd be a great way to cheat, right? Like you'd certainly be successful if you did that. The problem is it's also very dangerous because it makes your blood a lot thicker than it's supposed to be. And the viscosity of your blood can affect your blood pressure. And that can lead to things like ventricular aneurysms, strokes, okay, and things like that that can drop you dead in seconds. Okay, so you're taking a big chance with your health by doing that. So you got to decide whether winning the Tour de France is worth it or not. Oh, a week. Yeah. No, steroids are hormones that allow your body to repair the damage to the muscles that you do by training. Okay? When you train, you break muscle fibers, and your body rebuilds them. When it rebuilds them, they're stronger. Okay? So if you're taking steroids, you can essentially train twice as much as a person who's not, because it takes twice as long for your muscle fibers to repair themselves. Okay? So yeah, you can gain a lot faster if you're on the juice. Not usually a clotting problem, no. Because what they usually do is they don't just inject all of the blood, they take the fluid out, the plasma, and they just inject the red blood cells back, right? So it's a much thicker solution. Probably you would bleed less, I would think. I don't know for sure, but it would seem that you would probably bleed less because your blood would be thicker. You would have more clotting elements in your blood, and clotting elements don't typically activate until they're exposed to air, right? And that's when they activate. So, all right. So we got some examples here of single-celled and multi-celled organisms. Okay, you've got actinopods here. These are a marine single-celled organism. They make a shell that's actually silicate. It's glass. Okay, they make this shell that surrounds them. Okay, this is a paramecium. Lives in pond water. Okay, or sloughs, or even sometimes big puddles. Okay, and obviously you got a cactus here, which is a multicellular organism. The thing they have in common: they are made up of cells, whether that's one cell or millions of cells or what, they're made of cells. Okay. Okay, second point of the cell theory is the one that we come back to most often. Okay? Cells carry out the basic functions of the organism. Okay? That means essentially I could take a cell out of your body and put it on a petri dish that has nutrients on it, and that cell would survive without you, because it can carry out all the functions of an organism by itself. Okay? Everybody follow me there? I guess if I kept giving it stuff, theoretically it would keep going. It would reproduce, right? And produce more of itself and grow. Okay? And that's how, if you've ever had a throat swab, anyone ever had one of those where they stick the Q-tip in your throat? Okay? Yeah, and then what they do is they take that and they smear it on a petri dish, which has this stuff called agar, which looks like Jello, but it doesn't taste like Jello. Okay, it's got the stuff needed to make proteins. It's got sugar and all that, and bacteria love it. It's great, and they can grow on it, and then you get these colonies, and then they test the colonies to see what bacterial infection you have. All right, if I was to put one of your cells on there, it should survive. Okay, it doesn't always work that way, but theoretically, okay, any cell should be able to survive on its own because any cell in your body carries out the basic functions of the organism. There are exceptions to that, obviously, because some of our cells are so specialized that they've actually lost some of the abilities, but in general, this holds true. No. Uh-huh. No. No, it would grow the tissue. That's the idea behind stem cells. Okay, if I take a stem cell from my body that's well, I couldn't take it from my body because I'm already an adult. But if you took fetal stem cells, okay, and that's the one that we generally don't like because it means the fetus dies. Okay, if I take fetal stem cells, the ones that are supposed to become the fetus's liver, and I put them on a dish, I'll grow liver tissue. I won't necessarily grow a liver, but I'll grow liver tissue. Okay, and that's the idea behind stem cells is if I take a stem cell and I can grow liver tissue, I can graft that tissue onto someone whose liver is not functioning properly. Okay, or I can do that with a pancreas. Right, if someone has diabetes, I can take stem cells and grow pancreatic tissue, and I could cure diabetes and now they have pancreatic tissue that functions properly. Okay, things like that. All right, that's the idea behind it. Caroline. Huh? How much are they? I don't know. They're just little pieces of plastic. They're just a plastic dish. It's the agar. You got to make it. It's a mixture. I've never seen Petri dishes at Costco. Cam, question?
I've read like on the science magazine or something, there was a cell that was kind of like immortal because it kept reproducing itself and kept
Yeah. There's certain research that's going on into trying to make cells that essentially can live longer. Cells have a certain lifespan. Your cells have a certain lifespan based on the number of times they divide. Certain parts of your DNA get smaller each time a cell divides, and eventually, as they get smaller, it starts making more and more mistakes, and those mistakes lead to tumors and cancer and stuff like that. Yeah. I don't know much about that, honestly. I've heard that they're trying that stuff, but I'm not sure how a 3D printer makes a living cell. Yeah. Okay. It's, after all, just a printer. It's inanimate, right? Because they divide out of control. So a cancer cell, it's the reason that cancer is so hard to get rid of is it's your cells. Your body doesn't recognize them as a problem. They come from you. But when they divide out of control and they divide really fast, they make a mass. And that mass starts taking more and more of the nutrients that the healthy part of the organ is supposed to be getting. So your own cells actually end up starving the healthy cells, and that's kind of how cancer gets you. Okay, is these cells, and then they can get into other places in your body, and yeah, generally how it works. That's a really layman's explanation, but it's generally how it works. Sort of. Okay, the true answer is sort of. You're not really curing it. That person is still genetically diabetic, but if they have pancreatic tissue that functions properly, they produce insulin, right? Again, I don't know much about the use of the 3D printer for medical purposes. I know I've heard people do that, but I've never seen it done. I don't know anything about it. Okay. I guess. Yes, some organisms can regrow things. If you cut a worm in half, it'll grow back. Yeah. You rip a starfish into pieces, and each arm will grow a whole new starfish. Oh, yeah. Simpler organisms tend to have more of an ability to regenerate. Okay. All right, guys. I'd love to keep answering all these questions, but I got to get through some of this stuff here. Okay. Okay. Third point of the cell theory: new cells are not created. You do not have a factory in your body that produces cells. All the cells in your body come from the division of cells that already exist. Okay. That goes all the way back to the moment you were conceived. Okay. The moment you were conceived. One sperm met one egg. That made one cell, believe it or not. Okay, it was two cells, but they go together. They make one cell. Every cell you now contain is a descendant of that original zygote cell. All right? And when you produce more cells, they come from the division of existing cells in your body. You make blood cells by bone marrow cells dividing and producing more blood cells. Right. Well, we're not 100% efficient. You waste energy all the time. That's why you give off heat. I don't see how that has to do with energy. You and I consume food. That's where we get the energy to do stuff. No, it's not 100% efficient. It's not. No, you can't. It can't be more than 100% efficient. I don't know where you're going with that, Cam. I'm sorry. Guys, I got to get through some more of this. I'd love to answer all your questions, but I don't have time today. Okay, I just got to get through enough of this here. Okay. All right. So this is showing here. Okay, when your cell is going to divide, it's got to copy all of its DNA. Okay, that's the first thing it's got to do because the DNA tells it how to do everything. So it copies its DNA and then splits down the middle, and then you got two new cells after that. Okay, so all the cells in your body come from the division of existing cells. All right, so development of the cell theory. Important people. Most important guy, Hooke. Okay, oddly enough, Hooke is a physicist. Okay, he's actually more more well known for his accidental discovery of the cell. Okay, he actually studied like spring forces and things like that. Okay, he studied spring forces, waves, simple harmonic motion, stuff like that. This was actually a little experiment he did to test the lenses and the refraction of light. It just so happened that it magnified stuff. Okay, and he completely by accident discovered cells. Right? What he was looking at was a thinly sliced cork. Okay, so a cork from like a wine bottle. Okay, he had taken this cork and he had sliced it really, really fine and placed it under the microscope, and he found that it looked like it was made up of little rooms, like the cells in a prison or the cells in a monastery, whichever one makes you happier. Okay, cells are small rooms. Okay, now cork is dead, so they looked empty to Hooke when he looked at them. Obviously, we know now cells are far from empty. Right? But these ones were empty because they were dead. So all he saw actually was the cell walls of the plant cells from the cork tree that this cork was harvested from. Right? But he was the first person to discover cells. All right. Then this guy came along, Anton von Leeuwenhoek. Okay? Leeuwenhoek had this weird-looking microscope. Luckily, it didn't catch on because it would have been really, really, really hard to use. Okay? Essentially, you had to let light come through the lens, or sorry, light go through the object, so coming through this way, okay, then through the lens, and it would project an image. So if you held it far enough away, you could kind of see, or you could project it onto a screen maybe, okay, and kind of blow things up that way, but it would have been really, really hard to use, hard to focus, hard to hold still, okay, all of that kind of stuff. But it worked good enough for him, okay, he was able to see living cells. He was the first person to look at living cells. Hooke's cells were all dead. Okay, this was living cells. He could see that they functioned. He could watch what they did. He could see how they reproduced. Okay, all of that kind of stuff. And he found that basically everything he looked at had cells. Right? He looked at pond water. He found little, one-celled organisms, parameciums, amoebas, things like that. He looked at his own blood. Okay, he got sperm cells from a bull. I don't know how, but he did. Okay. And his microscope magnified up to 300 times. All right? Science can be a dangerous thing sometimes, I would suppose. Okay? All right. Then these two guys came along. These two Swedish fellows, Matthias Schleiden and Theodor Schwann. Okay? They took Leeuwenhoek and Hooke's work and they expanded on it. Their hypothesis was this: if cells are the basic unit of life, and tissue from all kinds of living organisms are viewed under a microscope, then cells should be present in all tissue. Okay? Years and years of experimentation and looking at stuff under a microscope proved that to be true. Okay? So they're the ones that actually proposed the original cell theory, that all organisms are made of cells, that cells carry out the basic functions of the organism, and that cells reproduce, okay, from the division of existing cells. Okay, now looking at cells has allowed us to do a few things. The really important things it's allowed us to do is find similarities between organisms that might on the surface not look at all the same. Okay? If I was to look at the cells of an earthworm and the cells of a human being, they would look very much alike. Okay? They have very similar structures. Now granted, if I'm looking at the same kind of cells, right? If I'm looking at, let's say, nerve cells from both, the nerve cells would look a lot alike. If I'm looking at cells from the digestive tissue, okay, they're going to look very much alike. Why? Because we descended from similar organisms, okay? We are alike. We're both animals. We both consume other organisms for food, okay? Things like that, all right? And so we have to have a fair amount of things in common, all right? So that's the big thing about cellular biology, is it allows us to find relationships between organisms we think may or may not be related. Right? Now, if I look at the cells of a fungus and the cells of an animal, I'm still going to find a fair amount of things in common. If I look at the cells of a plant and an animal, I'm still going to find a lot of things in common. Okay? That tells us something. If we've got a lot of things in common with organisms that really don't have a lot in common with us, there must be something to that. Okay, that something is that cells have to be the unit of life going way, way back. Okay, way, way back. So we're talking like billions of years ago. The first organism that appears on Earth has cells, and they have similar structures to what we see now. Okay, natural selection says if it works, we keep it until something better comes along. All right, cells in all the organisms that we see have very similar structures. Okay, that means they've been shared somewhere along the way with a common ancestor. All right. Okay, skip that one. Okay, so we look here. We got an animal cell on the left, got a plant cell on the right. Do they have a lot of things in common? Yeah, a lot of the structures are the same. There's more the same than there is different. Okay, they both got a nucleus. Okay, they both got this stuff here, the rough ER. They both have the smooth ER. They both have a Golgi apparatus. Okay, they both have mitochondria. That's these sausage-looking things. Okay, they both have. Okay, well we got the mitochondria there. They both have a cell membrane. Okay, right in here. They both have cytoplasm. That's the fluid that fills up the cell. Okay, not only is it structures, but it's also processes. Okay, the way plants and animals burn sugar for energy is the same. Okay, that says a lot about where we came from, where plants came from, okay, and kind of how life may have come to be, okay, on planet Earth. Now, I'm not trying to tell you anything different from religion class, and one of the lessons in this unit is me talking about how science and religion can get along. Okay, oh yeah, totally, man. Okay, science and religion are not the mortal enemies that you've been led to believe, I promise. Okay, and I'll explain all why one day, but not today, because I don't have time to do it today. Okay, but I will go over all that, and then you can all be happy again. Okay, because science and religion can be friends. All right. Okay. Well, no, really, some of the best scientists were actually men of the cloth. Okay, men of the cloth, like monks, priests, things like that. Some of the best scientists were. Okay. All right. So essentially what we have here is that they have a lot of things in common, okay, and it's because of those similarities that we can trace the lineage of living organisms to extinct or ancestral organisms. All right, microscopes. Real quick here. Okay. When you are using a microscope, keep in mind that you are using a delicate, expensive piece of scientific equipment. Please treat it as such. Okay? These have not been treated terribly well, and as a result, we have had to MacGyver up ways to keep them running, like this plug thing that we have on this one. All right? Now, part-wise. Okay? Actually, let's talk about handling techniques. This is the arm right here. Somewhere along the way, someone's telling you it's the neck. I don't know who that is, but they're wrong. Okay? This is the arm. Okay? It's the arm. This is the base. When you carry a microscope, you have one hand on each. Okay? It is not a bowling ball. Okay? Because I see people like carrying it like this. Okay? That is not how you carry a microscope. All right? You will drop it eventually if you're doing that. So one hand on the arm, one hand on the base. Okay? Carry it always with two hands. And make sure that the cord is wrapped up. Okay? And that you have it. Because I've seen this happen too. People are walking along, cords on the ground. They step on the cord. They keep going. And it pulls the cord out of here. That's hard to fix. Okay? The microscope is essentially a write-off at that point. Okay? So we don't want to have that happen. Okay? Prime example. Okay, this one's already had that done. Luckily, it pulled out of here, and we can fix that part. Okay, so we always want to make sure we've got the cord in there. Okay, the top piece here that you look through is the eyepiece. Okay, the ocular lens. Okay, is its technical name here at the top. You have your objective lenses here on the revolving nose piece. Okay, that's what this part, because it revolves. Okay, now the least powerful lens is this little red one. Okay, we call it the scanning power lens. Okay, because we use it to scan the slide until we can find what it is we're looking for. You're not going to see a lot of detail on the scanning power, but you'll be able to find what you want. Okay, once you've found it, you can use these little knobs here to move the stage around. Okay, you do that, and you get whatever it was you were looking at centered in your field of view. Once it's there, you can move up to the next magnification, and what you were looking at will still be there. If it's not in the center, when you move up, the amount of area you see shrinks, and what you were looking at may no longer be there. All right, so it's important to know that we have to be aware of that. Okay, we get it focused, we get it centered, we move up to high power, and we view it on high power. And that's generally where we do our drawings and where we see the most detail, okay, things like that. All right, there's two focusing knobs on the microscope. Okay, there's a coarse focusing knob, that's the big one, and a fine focusing knob. The coarse focusing knob, as you can see, moves the stage, this flat part, quite a bit with very little turning. The fine focus knob hardly moves it at all. That's important to know because if you're on high power, you do not want to use the big knob. Reason being, you can actually physically drive the slide through the lens. Sorry, the lens through the slide, other way around. Okay, and then that'll crack the slide, it'll scratch the lens. Okay, you'll hear the cracking sound, then you'll hear the thumping sound, then you'll hear the ambulance sound. Okay, so don't use the coarse focus knob on the high power lens. Okay, please use just the fine focusing knob. If you use the coarse focus knob, you're going to go right past the focused point anyway because it moves way too fast to be effective on high power anyway. Okay, and then you've got down below here the ability to adjust light levels, okay, and contrast and things like that with the condenser lens and condenser diaphragm down below. Okay, so those are kind of the parts of the microscope. Okay, once in a while you may need to clean the lenses. Sometimes there's a fingerprint on one of them and you can actually see it when you're looking through and it's really annoying. Okay, you use special lens paper to clean the lenses. You do not lick your finger and do this, okay? Nor do you take your shirt, okay, and rub on the lenses because you don't know what kind of really small grit might actually be on your finger or your clothes. Really small grit that you don't even notice makes a really, really tiny scratch on a microscope lens. What does the microscope lens do to the tiny scratch? Makes it bigger. Now the microscope is completely useless because all people can see is the giant scratch, okay, down the lens, so we don't want that. All right, so always use the special lens paper for that. Okay, always start with the lowest power and move up from there. Okay, so here's what we're looking at. If I use the scanning power lens, you're going to want to draw this. Okay, if I'm looking through the scanning power lens, I can see a circle with a diameter of 4 millimeters. That's 4,000 micrometers. Okay? So let's say I'm looking at a tiny little organism, okay, and it's over here. Okay, and that's all that I can see on scanning power. If I move up to the next power of lens, oh, that's not very good. Okay, if I move up to the next power of lens, do I see that now? No, right? My field of view on the next power of lens is quite a bit smaller. Okay, and so I won't even see that thing because it won't be in my field of view. Okay, and that will be counterproductive. Right? So on low power, okay, it's 100 across, or sorry, it's 100 times magnification, and the field of view is 1,600 micrometers. Okay, so from here to there is 1,600 micrometers on the next power up. So on scanning power, what you look at is 40 times bigger than normal. On low power, it's 100 times bigger than normal. On high power, it is 400 times bigger than normal. And that the circle that you see is only 400 micrometers across. It is 1/10 what you can see on the scanning power. So while you see more detail, you don't see as big a space. Okay? I always compare it like this. Let's say I'm like at a Flames game, all right, and I'm trying to find somebody on the other side of the arena, and I know that they're wearing a bright yellow shirt. Okay? Am I going to take out a telescope and just pan along looking for a bright yellow shirt, or am I going to look for a bright yellow shirt with my eyes and then put a telescope to my eyes? Which one makes more sense? The second one. I should first find them when I can see lots. If I'm trying to find one single needle in the haystack, okay, with this telescope, I'm never going to find it because I can only see this much area with the telescope. But if I'm using just my field of view, my eyes, I can see quite a bit more. I can find the yellow shirt and then zoom in on the yellow shirt with a telescope after that. Everybody follow me there? Okay, that's why we want to start always with the scanning power lens. Okay. All right. Now, if I want to estimate how big an object is when I'm looking at it under the microscope, these numbers become important. Okay? So I'm looking at this thing under the microscope right here that I drew. Okay? Let's say I'm looking at it on this power. So what, maybe, let's say 10 of those could fit across. How big is each one then? Each one is 400 micrometers. Okay? I estimated how many would fit across, and I divided how big the area was. Okay? It's 4,000 across. I think 10 fit across. Each one must be 400 micrometers in size. Okay? Estimating the size of the object is pretty easy. You just generally estimate how big you think it is based on how much of it fits across. If you were looking at high power and it took up half the field of view, how big is it? 200. Okay? Is that very hard? Okay? Estimating the size of an object is easy. And it is, after all, just an estimate. Okay? Thing to remember about estimating the size of an object, you always estimate on its longest dimension. All right? So if you're looking at something that is obviously longer one way than the other, always estimate on its longest dimension. All right? Okay, so that covers what we need to know about that. Those are the sizes if you didn't get them on your other drawing there. 4,000 micrometers on the scanning. Okay? And we're good. Okay, good. We got it all done. No quiz tomorrow.